A very good afternoon, everyone. So in the next few minutes, I will be telling you how molecular markers are used for timber identification. Now, by molecular markers, we mean DNA markers. And when it comes to identification of timber, the most important questions that we need to answer are from what species the sample has come from. And apart from that, other questions that need to be addressed are from where was it, meaning the geographical area from where that sample has come, or from which individual exactly, if it is from a particular species, which individual exactly does that sample in question belongs to. And sometimes questions arise as to what part of the sample it is also, whether it was the heartwood or the sapwood or the bark. And other questions like what is the age of the timber, and what was the period before which log was felled, and sometimes even about the inheritance also question needs to be addressed. And earlier, as Dr. Sinha had also mentioned, there are different techniques used for timber identification. And in order to determine the taxonomic, geographic, and individual sources of that timber, um, there are three kind of approaches that are available, are followed. The visual approaches, where most importantly wood anatomy plays the role. And then there are chemical approaches, and then we have the genetic approaches. So what I'll be concentrating is on the genetic approaches available for timber identification. And as sir had also mentioned earlier, here I would just like to point out that among all the questions that need to be answered when there is a timber sample that needs to be identified, uh, different techniques can be used to answer different questions. Whereas when it comes to genetic analysis, we are able to answer questions as to which genus or species our sample belongs to, and even the provenance, the geographical origin of the sample, and whether it is from a wild or cultivated population. And we can also narrow down to which individual the sample has come from. And of course, the age and the period after felling cannot be determined using genetic analysis or using DNA markers. Uh, Plant materials, or say timber, it can be identified by all of these techniques when it comes to species identification. But in the traditional methods, when the morphological characters are not present, then that is when the specimens are degraded, when they lack physical features. For example, if it is treated wood or modified wood, or sometimes even if it is powdered wood or a wood product, at that time identifying what species it is by the traditional approaches like wood anatomy becomes difficult. At that point of time, genetic analysis comes handy. And genetic analysis, most importantly, they use uh, the universal barcode system, or sometimes even DNA sequencing and other biomolecular techniques, which I'll be explaining. And these DNA markers can be used for identification of those specimens. Among the different genetic analysis techniques, karyotyping or chromosomal banding for plant identification is the oldest technique. However, this technique uses the principle of staining and visualizing the chromosomes of that particular species at the mitotic division phase. And since fast dividing mitotic cells are not available in timber, this is not a viable technique for timber identification. But I've just mentioned this here to say that this is the first genetic analysis technique that was in place for plant identification. And coming to the techniques that are used for timber identification, the other genetic analysis techniques are, the first is profiling or fingerprinting markers. They are actually DNA fragment markers, where the DNA is fragmented And the fragments are analyzed after agarose electrophoresis, which produces a banding pattern when visualized under UV. And based on the differences in the banding pattern, we are able to, and that difference in the banding pattern is called the fingerprint as far as DNA markers are concerned. And based on the difference in the banding pattern, identification is done. And there are different techniques in DNA fragment markers. And the next set of markers are DNA sequence markers or barcode markers. And these technique uses specific sequences within the DNA sample of that species for identifying. And the profiling markers can identify samples at the population or individual level also. And barcoding markers can be used for identification of taxonomic levels like the genus and even up to the species. And when it comes to DNA profiling markers, they are basically based on PCR or polymerase chain reaction, which amplifies a particular segment of the plant DNA. And when that segment is amplified meaning it is produced in many copies, and that is electrophoresis in agarose gel. And when that is visualized under UV, this kind of a banding pattern is observed. And each of those bands, using standard molecular weight markers, the molecular weight of those bands can be determined. And because of the difference in the molecular weights, this differences in the banding pattern happens. So each of those samples, each of the samples that we study will have a different banding pattern because of the variable size of the amplicons produced during the PCR technique that is followed. And using the principle of PCR, different DNA molecular markers have been are being used. This is RFLP is restriction fragment length polymorphism. This is the only profiling technique that does not use PCR actually, where it uses a principle of restriction of the actual DNA sample that we have. The complete plant sample DNA is actually broken into smaller pieces using restriction enzymes, and these restriction enzymes are site specific. They bind to a particular site in the DNA and cleave them at that site. Site specific meaning a particular nucleotide sequence has to be present. Only if it recognizes that nucleotide sequence, it is able to cleave or cut the DNA at that point. And wherever that specific site is available, everywhere a cut is made, and then different fragments are produced, each of them having a different molecular weight. And DNA samples, they can be visualized only by gel electrophoresis. So agarose gel electrophoresis is performed, and then we are able to differentiate samples. And this was the very first DNA profiling technique that was used even before PCR was invented. And after that, in combination with the PCR, PCR RFLP techniques are being used, where amplification of particular fragment of the DNA is done, and then restriction is done, producing variable size DNA fragments for analysis. So during this restriction fragment length polymorphism, what happens is, in different samples, when the binding site for the enzyme is available, for example, in sample A, the binding site is available, so the enzyme is able to cleave the sample at that point. Whereas in sample 2, the binding site is different. That is, the DNA sequence, the nucleotide sequence is different. So the enzyme is not able to recognize that site, and then it does not cleave at that site. So the sample 2, a single, when electrophoresis is done, in sample 2, a single band is observed, whereas in sample 2, because of that fragmentation, two different DNA fragments of different molecular weight, so two bands are observed. So this is how different samples, they will produce a different banding pattern. And this is when there are more than one restriction sites or restriction sites at different places in the same, at the same region of our samples. So each of these samples, if you are able to see, like in sample 1, in near the 5' end only a cleave is made. So a longer and a shorter fragment is made, so the binding pattern is different. Whereas in sample 2, there is no site, so the longer fragment is present. It moves very slowly during electrophoresis and it remains up in sample 2. Whereas in 3, compared to 1, again one cut is made, one cleave is made, but it is at a different site. So 2 fragments are generated. And in sample 4, 2 regions in the same sample cleave is made, resulting in 3 fragments. So this is how polymorphism is exhibited in the DNA fragments that are obtained, and then samples can be differentiated. And after that, the other DNA profiling techniques are all based on PCR, all polymerase chain reaction, where specific segments are amplified. Now, this RAPD, or randomly amplified polymorphic DNA, this uses universal primers, primers which can bind anywhere to our DNA samples, and wherever the site for binding of these primers are available, they bind, and then the regions between the forward and the reverse primer is amplified. This is a single PCR reaction, and even up to individual identification is possible using RAPD technique. And this is how it works. Like in sample A, at the different positions, at 4 different points, it is able to, the forward and reverse primers are able to bind. So 2 fragments are generated. Whereas in sample B, at 1 point for the forward and one at 2 different points for the reverse, it is able to bind. But again, the locations where the primers bind to our DNA sample are different. So only 1 fragment is generated. And again here, the fragments are produced after PCR, that is after amplification of those regions. So each of these fragments, they differ in their molecular weight, and after gel electrophoresis, they can be visualized by staining with ethidium bromide, and based on fluorescence, they are visualized under UV, and these bands, changes in these bands can be used for identification. And the other is AFLP. So in this technique, this again, it uses the PCR technique, but apart from the primers that we add, we use certain adapter sequences also to bind to our this polymorphic, the polymerase chain reaction. And then different sets of regions are amplified, but this is not a single PCR reaction. Different reactions are carried out. And during AFLP profiling also, even among closely related individuals, individual identification up to individual identification is possible. So what happens actually during the AFLP is amplified fragment length polymorphism. What actually happens is, first the DNA sample from our plant is restricted or cleaved with the different restriction enzymes, and then adapters are ligated or joined to those fragments. And wherever these adapters bind, again that binding is also random. These adapter sequences are used for developing primers, and since the total sequence, DNA sequence of our sample might be unknown, we don't know what species it is, and we may not know which regions can be amplified during the RAPD also. So we use adapters, and then sequences complementary to the adapter region are used as primers, and then first one pre-amplification is done, and after that we can do selective amplification during the second set of PCR. So when a selective amplification is done, again our primer sequences differ, and then when you do electrophoresis of the fragments that are obtained, here a large number of fragments are obtained, and even between closely related individuals, even if there are minor sequence differences, they will also be reflected during AFLP. That is how individual identification is possible using this technique. So this is how it works in the normal PCR also. The whole genomic DNA. Of course, for all these to use all the DNA marker based techniques, the DNA has to be first extracted, and then it is restricted with a specific enzyme, and adapters are added. These adapters, they bind to the double-stranded DNA because of the cuts where the restriction enzyme makes. The cuts where the restriction enzyme makes are also known to us because each restriction enzyme binds only to a specific region in the DNA. And based on that, these adapters are added. And then after that, we see that ligation happens after the restriction and ligation. Then we do selective amplification using our primers. And this selective amplification can also be based on the sequence of the adapter that we have added, or based on the cut where it has where the restriction enzyme has made.